to study microbes, particularly wine microbes in our case. And so it's broken down into classical techniques, microscopy and culture, and the more modern molecular genetics and genomics approaches. So I'd like to start off with the classic gram stain because it's still used widely in microbiology labs, including wine microbiology labs. So in this particular case, you begin with flooding a smear, heat-fixed smear with crystal violet, which is a basic cationic dye that will stick to negatively charged bacteria uniformly. In step 2, you're using iodine, which is a mordant that increases the affinity of crystal violet for the cells, so everybody continues to be purple after this step. In step 3, this is what distinguishes gram-negative and gram-positive, and so the thicker peptidoglycan layer of gram-positives resists the penetration of alcohol, whereas the thinner peptidoglycan of the gram-negatives allows the alcohol to come in and wash it away. And then the last step, you counterstain with safranin so that these gram-negative bacteria will take up this basic cationic dye and stain a light pink. And I have a video for this posted as well, so you can watch the gram stain video. And hopefully you remember doing it in micro 221 or 224. So the net result is, in this particular case, you see a mixed bag of gram-positive and gram-negative bacteria, and so you can look at morphologies and relative proportions of gram-negatives and gram-positives. Over here is just a gram stain done on a population of Saccharomyces yeast. So again, they stain gram-positive technically because they take in the first dye, crystal violet, and largely resist decolorization based on their cell chemistry. So they're not technically gram-positive in the sense of like you would call a gram-positive bacterium gram-positive. These guys are just going to take the first dye and hold on to it no matter what. One thing we also want to do is measure cells. Sometimes measuring cells gives us some important information. Size of the cells can be important information. And so each microscope that we have is fitted with an ocular micrometer that you can see through one or the other ocular. In order to get a sense for how big these spaces are between the lines, we can lay down a stage micrometer and then focus in on it and then align these. And so the stage micrometers have fixed distance between the lines. So in this example, the fixed distance is 10 microns. And so we can line up these larger lines, and then we would know, for example, that each individual gap is 1 micron because 10 of these equals one of these spaces, which is 10 microns. So obviously 10 divided by 10 is 1. I should go back to this and just mention I do have a video for this, and so you please watch it and make sure that you understand how this is actually done. The next thing I wanted to point out was live dead staining using a hemocytometer. So hemocytometers are a great way to quantify microbes in a sample. They're especially good for quantifying yeast in a sample. But when you add methylene blue to the sample, you can actually distinguish between live and dead cells. So this is really useful when you're experiencing a stuck fermentation, for example. You can look at the cells and not only look at whether or not they're budding, but you could also look at the proportion to live and dead cells. And so methylene blue in its oxidized state is blue, but methylene blue in its reduced state is colorless. So if you collect some yeast cells from juice or a pure culture and then treat them with methylene blue, the live cells will take in the dye and reduce it to a colorless form. The dead cells, however, the methylene blue will just stay oxidized. And so again, you can count the number of bacteria or yeasts in each small square on the grid and then take an average of that and use this formula here to calculate direct microbial count per mil. So in this particular example, assume that an investigator counts an average of 20 microbial cells in a volume of 1/16 millimeter squared by 0.1 millimeter deep. The DMC per mil can be determined using this. So the average number of cells in the smallest square. So again, taking the total number of microbes in each of these 4 squares and then averaging it would go here. The reciprocal of the area of the smallest square. So here's our area of the smallest square. So we want the reciprocal of that, 1/116. and then the reciprocal of the depth, which is 1 over 0.1, times 1,000, which is our constant because we want to convert millimeters to centimeters, and then the reciprocal of any concentration factor. So if you've concentrated the sample, in this particular example, there is no concentration or dilution, but imagine if I do throw that at you in an exam, the reciprocal of any concentration factor. So if you've concentrated the sample, then you have to divide by that number in order to get the true value of the sample, because otherwise your answer is going to be a reflection of the concentrated value as opposed to the actual value. Similarly, if you do a dilution where you've diluted the thing down quite a bit, you have to multiply back up by that dilution factor to get to the original. And again, there's a video on that, so you can get a sense for how to do these calculations as well. And obviously, I'll answer any questions to help you. One of the other versions of viability stains are fluorescent viability stains using Fun1 dye and Calcafluor. So Calcafluor is not a viability stain. It's actually just a general yeast stain that we often use, and you can see why it's so useful. It gives you these nice halos around the yeast cells that allow you to quantify them or take beautiful pictures like this. Whereas Fun1 dye actually penetrates the yeast and will give you a distinction between live and dead yeasts through the formation of these little things called sieves. And so the these sieves are these orange particles that form inside vacuoles, inside live cells. And so this is a brief video of one that I did. So here's a live cell, and you can see the sieves bouncing around in this vacuole. Anybody who has a vacuole with these sieves in it is considered viable, and anybody that doesn't is not viable. It's a very expensive dye, so I don't use it as frequently in class anymore, but we found that methylene blue works really well. So some of the plating methods that we use in a typical wine microlab include membrane filtration. So you could, for example, take a 750 ml bottle of wine and run it through a small pore filter, capture what's on that filter, and apply it to different plates to do plate counts of microbes in that wine. So if you get a bloom of microbes in a case of wine, you can pull out a bottle and crack it open and run it through a filter like this and perform this type of total plate counting. Another thing that can be done, this is mostly in terms of sanitation, in bottling rooms and so forth, you can do Rodak sampling for surfaces. So again, these particular plates, the agar is poured so that it forms sort of a convex layer over the top, and then you can stamp that down on a surface. And the basic formula that we use then is, knowing that the Rodak plate has an area of 4 square inches, we can then stamp it down, get some colonies to form, and then count the number of colonies to get an estimation of CFU per square inch. Knowing again that the plate is 4 square inches, if you get 50 colonies to form, 50 divided by 4 is 12.5 CFU per square inch. We don't want more than 50 CFU per plate. That's considered unsanitary. But obviously you have to consider what the microbes are. So if you get less than this, but all the microbes are salmonella or something terrible, then obviously that raises an alarm too. You can't just say, oh, cool, it's less than 50. But if it's less than 50 and it's all listeria or some terrible microbe, then that's not a good thing. The spread and streak plate involves spreading mixtures of cells on an agar surface so that individual cells can be well separated from each other, and then they can form separate colonies that can be quantified. So the spread plate technique looks like this, where you basically dispense cells into a medium, onto medium in a petri dish, and then you take usually we use a glass rod or a metal one, and dip it into some alcohol, and then light the glass rod on fire, let it cool, and then spread the microbes around the surface of the plate. If done properly, if your dilutions are proper and your technique is proper, you should get a good distribution of bacteria and you should be able to quantify individual cells. This one happens to be nice in the sense that it's got yellow and white colonies that are easy to visualize. So again, these approaches, this spread plate that you see here, is great for quantifying microbes in a sample. The streak plate, on the other hand, is a way to rapidly get isolated colonies, but you can't quantify. You can quantify individual isolated cells, but you can't quantify the amount or the concentration of microbes in the original sample, because obviously all this is smear. Unlike this, where you could quantify these colonies and then backtrack to the original tube, and I'll go over that in a minute. But anyway, the streak plate technique usually involves dipping your loop, a sterilized loop, into a culture, making a smudge on the edge of a plate, flaming the loop again and cooling it, and then dragging microbes out, flaming and cooling and dragging microbes out, and then repeating that process of flaming, cooling, and dragging the microbes away from the original inoculation point. So ultimately, you get some isolated colonies. But again, this is not useful for quantifying the original concentration of cells in a culture, as opposed to the pore plate and streak plate, which are. So the pore plate, typically the sample is diluted many times, depending on what it is, and then the diluted samples can be mixed with liquid agar, and then the mixtures of cells and agar can be poured into sterile culture dishes. And so basically what you want to do, or what you might do in this particular case, is first perform a dilution series. So in my hypothetical example here, I have a concentrate here, an original sample. Imagine this is juice before fermentation or during fermentation, and you perform serial 1 to 10 dilutions. So 1 ml of the concentrate goes into 9 ml of diluent for a 10 to the minus 1 dilution. You mix that and then dip a sterile pipette in that, move 1 ml into the next dilution tube, mix it. That would be a 10 to the minus 2 dilution, or 1 to 100 dilution, and then continue along the line so that we end up with a 1 to 10,000 dilution. And then we have a 1 to 1,000 dilution, a 1 to 10,000 dilution at the end. So in our hypothetical example here, I could plate, either spread plate or pore plate, 1 ml onto this plate, and then spread plate or pore plate 1 ml onto this plate. Now, typically spread plates are done in much lower volumes. So in this particular case, this spread plate is typically a 0.1 ml spread plate, because if you pipette 1 ml into this plate and then try to spread it around and get that 1 milliliter of fluid to absorb into the agar, it's really difficult. So again, typically spread plates are done at 0.1 ml, whereas pore plates are typically done with 1 or 0.1 ml volumes. So if this were a pore plate, you would take the 1 ml out, put it into an empty sterile petri dish, and then pour molten agar on top of that. The net result here is that we get 1, 2, 3, 4, 5 colonies, and here presumably you can imagine should be around 50 colonies if this is a true tenfold difference between these two. So imagine this is 5, imagine this is 50. You can count that for yourself. I'm not sure what it is. But then we would use this formula to figure out the estimated CFU, or colony forming units, per milliliter. So using this hypothetical example and this formula, we can walk through the problem here. So C, the numerator, is the total number of colonies counted on plates having 25 to 250 colonies. So we'll imagine that we're not going to follow that rule just for the sake of walking through this problem. So if we're just going to straight up calculate C without paying attention to the 25 to 250 rule, then and we're also going to assume that this is 50 colonies, because I didn't pre-count it before I started this lecture. But anyway, imagine this is 5 and imagine this is 50. So C would be 55. So I would put my 55 here. My denominator is 1 times N1 plus 0.1 times N2 times dilution factor. So N1 in my hypothetical example is the number of plates where the counted colonies are from the lower dilution. So in this particular case, this is the lower dilution, and I've only done one plate. And so my thing would be 1 times 1. My N2, which is the number of plates at the next lowest dilution, is also 1. So again, that would be 0.1 times 1 times the dilution factor. And the dilution factor is of the lower dilution of your plate series. So again, that's this from this plate series. So my dilution factor would be 10 to the minus 3. So I would pop 10 to the minus 3 in here. And then you can just do the simple algebra to figure out the CFU per mil on that particular problem.